Thank you so much. It's a great pleasure to be here today. So, memory. Our memory holds our personal history. It allows us to change throughout life. In fact, much of what we know about the world is not built into our brains at birth, but it is acquired throughout life via our experiences. Our life story happens because our brains record and store our experiences. This is memory. Experience renovates the brain. This is how the brain records information. It is via this renovation. So, how do we know more tomorrow than we do today? This is because of this beautiful, brilliant organ, the brain. It is built to be adaptive. It is designed for change. It is built to renovate. The adaptive powers of the brain are due to what scientists call neuroplasticity. This neuroplasticity is allowing the brain to always be under construction. When we have a new memory, this is because the brain has been renovated. So neuroplasticity is a remodel for your brain. This is a neuron. These are the building blocks of the brain. This is one type of cell in the brain. You can see at the beginning of the neuron, those are dendrites. This is where information comes in. It goes through the cell body and the axon to the end, out at the terminal bouton. When multiple neurons are activated together throughout life, they form these neural networks, and these neural networks are what allow memories to happen. This is a synapse, the red circle there. This is the communication point between neurons. This is at the end of the terminal bouton. There's a cleft there called the synaptic cleft, and then there are the dendrites. That space in between in that communication point is considered a synapse. So the brain contains many, many networks of neurons. And our experiences, our day-to-day life, things that happen to us, things we learn, things we remember, reshape our neuron networks. The key to this neuroplasticity is growth and complexity of these neurons. This enriches neuron communication and supports new learning and memory. This is how new learning and memory happens. So neuroplasticity is key to our every being. It allows us to learn and remember. And in fact, our memories make us who we are. If you stop for a minute and you close your eyes, so everybody close your eyes for a second, and think about who you are, think about a memory. You're probably thinking, you can open your eyes now, you're probably thinking about a particular event, something that's happened in your life that has helped to make you who you are. This is an episode, and we call this type of memory episodic memory. Our memories make us who we are. These episodes of our life build and determine who we are. So how does the brain make memories? How do we make memories? It turns out that learning and memory are intimately linked. As scientists, we have to operationally define different terms. The way that a scientist like myself defines learning is that it is a long-term change in behavior as a function of experience. We operationally define memory as the ability allowing one to recall information that has been learned. Memory storage has 3 stages: the sensory register, the short-term store, and the long-term store. The sensory register, the initial portion of memory storage, is an initial site with a snapshot of the memory. This is very fast, 1/2 to 1 second. So an example of this might be if you look up a phone number and you need to call it, that immediate memory right there is that sensory memory. Short-term memory is where information is temporarily held. So for example, you write down a phone number and you're trying to remember it. So you say 727-0766, 727-0766. This is your short-term store. Long-term store are sites where information is stored permanently so that you can access it anytime, presumably, in the future. So the short-term store is often discussed in terms of short-term memory. As scientists, we talk about this typically in terms of minutes to hours. The long-term store is typically talked about in terms of long-term or reference memory. And again, this is information that is stored permanently. There's also a very important type of memory that allows us to navigate through our world and to update our world on a minute-to-minute basis, and this is working memory. This is information that is manipulated. So a way that I like to think of it is working with memory. You need to manipulate the memory. An example of this might be adding up the phone number. So for example, just remembering the phone number, 727-0766, is short-term memory. Manipulating this information, 7 plus 2 plus 7, that would be considered working memory. And people discuss this in terms of it being a type of short-term memory and probably occurring somewhere in between the sensory registry and the short-term store. So the process of making memories is called memory consolidation. There is an incredible amount of scientific literature trying to understand how memories are made in the brain. The brain is under construction during memory consolidation. This is when short-term memory gets converted and processed into long-term memory. So short-term memory, you start out remembering information, and it is in the hippocampus. We consider this to be the processing unit of the brain. Information gets consolidated, it moves out of hippocampus into the cortex, and we think of the cortex as the storage unit of the brain. So I'm giving you an example here. This is a human brain, and you can see the green strip right there is about where the hippocampus is in the temporal lobe. Information gets encoded and transferred to the cortex, which is the crinkled part there on the outside. And in fact, I have a sheep brain here, and I'm going to show you the hippocampus and the cortex firsthand. Okay, so here is a sheep brain, and here is the cortex. You can see it's the crinkled structure that is on the outside. This here is the frontal lobe. Working memory seems to be occurring in this part of the brain. Here on the side is the temporal lobe. Now, if we take a coronal section of the sheep brain, if we were to cut here, right here, and remove the front part, I've already got a dissected brain here to show you. You can see the hippocampus is in the middle. It's right over here. So information starts here, and it gets processed and moved out to the cortex. And this is consolidation. If interruption happens here during consolidation, for example, via concussion, via stroke, via extreme stress, long-term memories generally are safe. However, recent memories that have not been consolidated are at greater risk for loss. Those memories that have not moved out of the hippocampus and into the cortex seem to be at greater risk. So neuroplasticity is critical for consolidation. The brain needs this renovation to form new memories, to be able to learn new things. So when Dr. Colleen was talking about the law of connection as a behavioral neuroscientist, I jumped to this kind of connection, a different kind of connection. There are a lot of brain changes that occur with consolidation, and some of these changes include chemical changes, construction of new proteins. construction in specific parts of neurons, and construction in the connections between neurons. This allows for new networks to be created. Neurons change with learning and memory. Connections strengthen, dendrites grow and become more complex. So if I can draw your attention to the dendrites, those are the branch-looking structures there. You can see that when they grow, look at all the new synaptic points of connection here. There's an increase in these synapses. This is what I mean when I say the brain is under construction. So I'm a behavioral neuroscientist. I study memory. How do scientists study memory? There are a lot of ways to do this. One way is to study rats as they solve maze tasks. In fact, rats are able to solve quite complex tasks that require different types of learning and memory. Typically, rat and mouse mazes are defined by spatiality and by memory type. So what do I mean by spatiality? There are two types of spatiality that we talk about when we refer to rat tasks. One is non-spatial. This is utilizing intra-maze cues within the maze in order to solve the task. An example would be that first picture right there at the top, where one side is white and one side is black. The animals need to learn always go to the white side or always go to the black side, irrespective of space. So for a task like this, we flip the maze over, so the animal has to follow the cue inside the maze and ignore cues around the maze that are spatial. Spatial requirements use extra maze cues to solve the task. So those would be cues around the room. You all have spatial memory. You navigated here from your home because you knew to get in your car, draw up that cognitive map, and navigate to the Phoenix Convention Center. That is how you got here. That is utilizing a cognitive map in your spatial memory. In fact, that utilizes the hippocampus. Memory type is another way that we define maze tasks. Working memory, as I mentioned, is a form of short-term memory. For a rodent researcher, we talk about this in terms of information needing to be updated within a session for a particular maze task. I'll give you an example in a minute. Reference memory is a form of long-term memory. This is information that remains constant through all of the testing. One of the examples that I like to give for reference versus working memory for humans is where your parking garage is, wherever you park for work, your parking lot, your parking garage, that is reference memory. That information does not change. However, the information about where you parked your car in that day always has to be updated. You can think about this updating as working memory. You have to break the old association of where you were yesterday and figure out where the heck you parked your car today. And I say it that way because I get lost myself all the time. So the water radial alarm maze is one way that scientists study learning and memory in rodents. This is a spatial memory task, which means we require animals to use cues around the room in order to navigate through the task and solve it. It is working memory because information must be updated within a day. Those are the platformed arms. I will give you an example in one moment. And reference memory is a form of long-term memory, and this is information that remains constant across days. Those are the arms without the platform. So we drop a subject off, it swims, and it finds the platform. It remains on the platform for about 15 seconds. After this time, we remove the animal from the maze, and the platform gets removed. The animal needs to remember, I already went to that place in space, do not go back. If the subject goes back to this place in space, we consider this to be a working memory error. Working memory load, how many items of information an animal must remember across trials within a day, increases as trials progress. And this is because as animals locate more platforms, the load increases. So this is an example of data collected using mazes. The y-axis, that is the axis going up and down there, is working memory errors, and the x-axis is trial. And that first block there is the low load trial, the moderate load trial, and the high load trial. And remember, working memory load increases as trials increase on this particular task. So what is shown here is that the animals in purple, the young animals, were the same as the old animals on the earlier trials. On the middle trials, there started to be a little bit of divergence, whereas the aged animals, the orange there, started showing some impairment. And the very last trial block there shows the aged animals were markedly impaired. So this tells us many, many things. One of the things that this tells us is that aged rats are worse than young rats at handling an increasing memory load. But something else that this tells us is that on those early trials, those very early trials, trials 2 and 3, the young animals performed like the old. So, we got these data and we thought, wait a minute. So it's not that aged animals can't learn anything. They just need to learn it when it's a lower demand. So we took a look at the learning curves. And when you're studying learning and memory, this is critical because it tells us the way that the animals are declining in error scores, which is considered to be learning. So the y-axis here is working memory errors, and the x-axis on the bottom is days 2 through 12. Day 1 is considered an acclimation day. And what you can see is that for the low load and the moderate load and the high load, the young animals did beautifully. They've got great, great learning curves. But look at this. When we add the aged subjects onto this graph, we can see that they learned only with the low memory load. At this lowest load point, both of them learned. However, at the moderate load, their error scores are flat. They are not decreasing across time, and the same with the high memory load. So we interpret this as indicating to us that old animals can learn to handle only a low load, but not the higher loads, whereas the young animals can learn to handle memory at any of these loads for this particular task presented to us. And this is the way that we think about learning and memory studies from the standpoint of enhancing health overall, because now what this gives us is an opportunity to go ahead and give therapeutic interventions to try to make it so that the aged animals are showing enhanced learning and memory similar to the youngs. So I see my science in shapes and in colors, and in the last couple of years I've started painting my science just for fun. This is my first painting ever. It is called Dancing Neurons, and the inspiration for this is brain networks that are optimized. So this is a dynamic plastic system that is always under construction. It's a brain that is flexible and adaptive to external and internal changes. It's striving for homeostasis for balance. And dancing neurons are really the goal for a brain, right? We want a system that's in sync, responsive, adaptive, balanced, that has homeostasis. And really, don't we all want this for our lives? So my wish for all of you is that you have dancing neurons. So regarding the people that are doing the work in the lab, I'd like to acknowledge my graduate students, my postdoctoral fellow, all of the undergrads and the high school students, and my lab manager. Science is never done in a bubble or a vacuum. The only reason that I have had any success as a scientist in the last 10 years since I started doing this work in my own laboratory is because of my students. They work very, very hard, and they are really the scientists of the future. So maybe in a couple of years you'll see them here presenting as well. And then, of course, the funding, the National Institute on Aging, the Arizona Alzheimer's Consortium, the Institute for Mental Health Research, ASU CLAS, and the ASU Department of Psychology. And I also wanted to mention that we have just put out a book that I am an editor of, and I say we because it wasn't just me that wrote this book. This was a labor of love, not just for myself, but for many of the authors that contributed. So the name of this book is called The Maze Book, and it talks about learning and memory, a history of learning and memory, and how this work can lead to new discoveries in the field for health. Thank you very much.
Okay, well.